Good morning. It's Wednesday, April 6th. I'm Dr. Mario Castro, Vice Chair for Clinical and Translational Research and Chief of Pulmonary and Critical Care Medicine at KU Medical Center. Well, I'm not Dr. Steve Stites. I'm sitting in for Dr. Stites, and I think he must be enjoying some well-deserved time off, probably going around that corner on a canoe, looking at the next spot to go fishing. I want to introduce you and welcome you to our second Show Me the Science. If you're a regular viewer, you know we always follow the science. We also recognize that years of medical school and research make it easier for us to do that. We want to make it easier for you too. Today, we're taking a deep dive into the research around COVID-19 vaccines. By the end of this program, you'll be able to dispel some of those common myths around these vaccines and feel confident in your choice to get it, including boosters. Now, I have a lot of help today. Our panel of experts includes Dr. Gregory Poland, founder and director of Mayo Clinic's Vaccine Research Group, as well as editor-in-chief for the Journal of Vaccine. Welcome, Greg. Also joined by Dr. Christopher Harrison, researcher and pediatric infectious disease physician at Children's Mercy Hospital. Welcome, Chris. But first, Jessica Lavelle joins us. Do we have any reporter questions this morning, Jessica?
Good morning, Dr. Castro. Nice to see you today. Glad you're with us. We do not have any reporters on the line as of this moment, but we will get to our community questions here in just a bit.
Sounds good. Thank you, Jess. Now, vaccinations have been with us for well over 2 centuries. This really sets the stage for our discussion today. We've condensed those 200 years of vaccine research into 3 minutes for you. How many of these have you had or heard about?
Vaccines have been a part of protecting public health for more than 200 years. In 1796, the smallpox vaccine was developed from cowpox. The vaccine, through an unprecedented global immunization campaign, wiped out naturally occurring smallpox by 1980. Before 1885 and the first rabies vaccine, nearly all people infected with rabies died. 1914, a pertussis vaccine, more commonly called whooping cough, was licensed in the U.S. By 1991, the vaccine is expanded to protect against diphtheria, tetanus, and pertussis. The flu pandemic of 1918 lasted 2 years, killing more than 550,000 Americans and 20 million souls worldwide. It sparked efforts to develop a flu vaccine that was finally realized by 1945. But it wasn't until a second pandemic in the late 1950s, killing 116,000 people, that the U.S. Department of Public Health began recommending flu vaccines for at-risk people. By 2010, Dr. Gregory Poland with the Mayo Clinic recommended all healthcare workers be vaccinated against the flu. 1955 brought us the polio vaccine. 1963, measles. 1967, mumps. This vaccine took 4 short years to develop, and that record was held for 51 years until the COVID-19 vaccine. 1969, rubella vaccine arrived and was combined with measles and mumps to create the MMR shot. The first case of Zika was discovered in 1952, and by 2016, the virus was declared a public health threat by the World Health Organization. The threat ended later that same year when researchers, including Mayo Clinic, developed vaccines. In 2002, severe acute respiratory syndrome coronavirus, or SARS, sickened 8,000 people in 29 countries and killed 700 before disappearing in 2003. In 2012, Middle East respiratory syndrome coronavirus, or MERS, infected 2,500 people and killed 800. Researchers continue to study and develop vaccines for both viruses. mRNA vaccine technology for COVID-19 predates the SARS-CoV-2 virus by 36 years, when colleagues at Harvard University in 1984 used synthesized RNA enzyme to make messenger or mRNA in a lab. 3 years later, another discovery showed that mixing mRNA with fat droplets caused human cells to make proteins. By 1990, researchers were using mRNA as flu and cancer vaccines in mice. During the early 2000s, however, the lack of funding slowed the research. Still, in 2005, researchers made an important discovery when they learned that modifying synthetic mRNA kept the immune system from attacking the messenger RNA. mRNA research continued for 19 additional years until SARS-CoV-2 arrived in 2019. Scientists were ready. Money and manufacturing made possible in part through Operation Warp Speed fueled what medical scientists needed most to convert decades of research into COVID-19 vaccines and test them in clinical trials worldwide.
That was a great summary, Jessica. It really points out how important that research is behind the development of these vaccines. Now I want to turn to Dr. Poland for a moment. You've dedicated your life to vaccine research, including mRNA vaccines. As noted in that timeline, all along there was quite variable timelines between some vaccines and when they came out and when the infection started and when that vaccine came out. So, in general, how important is that funding in that research and the development of a vaccine, Dr. Poland?
That's a great question, and a very short answer is it's critical. Without that kind of research funding, without that long development pathway that you just very well illustrated, these vaccines would not be available to us. So, absolutely critical that kind of funding go to pharma, to academic researchers, to biotech. That's where we have generated and developed every single vaccine that we enjoy.
Thank you. And do you think that there was a difference here with COVID-19? It appears this really set the stage for our work here with COVID-19.
Very much so. I mean, as the video pointed out, work had been going on for decades. It's really a piece of misinformation when people suggest that these vaccines were somehow rushed or, developed with inadequate attention to the usual science. That's just not the case. These vaccines started being developed in earnest back in 1990. And actually had preclinical vaccines developed and going into animal testing by 2002, 2003. As the video pointed out, those were sort of shelved when both SARS-CoV-1 and MERS failed to really be an ongoing issue or epidemic. But that doesn't negate the fact that a lot of work had been going into the development of these vaccines. So what Operation Warp Speed did was, one, a recognition, an explicit recognition that we were in trouble, that this was likely to be a worldwide pandemic. This was likely to cost a lot of lives and result in an unmanageable surge demand on the medical system. We had technology on the shelf. And what Operation Warp Speed did is take a calculated risk. The government put billions of dollars on the table, research and development dollars, to look at the development of multiple platforms, not knowing which one would work best. Those included mRNA vaccines, the adenovirus vectored vaccine, of which Johnson & Johnson is an example of, and then recombinant protein vaccines, which we will see in the next month or two, and probably the exemplar for that will be the Novavax vaccine.
That's great. I hear that from my patients sometimes that are hesitant to get this vaccine. They're saying, this is really too rushed. I really want to know a lot more about this vaccine, and I give them that reassurance. I say, we've been working on this for now over 20 years, as you mentioned, and really this set the stage for us to be able to respond to this pandemic in a very quick fashion, but doing it the right way with good clinical trials to move the vaccines so we can get the shots into the arms of our patients.
Yeah, absolutely. And, I say this next statement by way of reassurance. There's nothing that you have ever been prescribed, no vaccine that you've taken that has had, with one exception, maybe rotavirus vaccine, that has had as many clinical trial participants and as much data gathered as quickly as has been gathered. Recognize that with our safety surveillance systems, we are catching side effects that occur as rarely as 1 in a million. I mean, that's by any stretch of the imagination. Up till now, that would be virtually unfathomable that we would be capable of that. So when you hear people saying that, that's sometimes a cry for knowledge. More often, it represents sort of fear-based thinking, because the data would not support such a contention.
That's a great point. And, as we do with Show Me the Science, we want to know about, how does this impact our children? And so, Dr. Harrison, why do vaccines for children lag behind the adult vaccines? We certainly didn't have them day one.
Dr. Harrison's muted.
Dr. Harrison, could you unmute, please?
Didn't know I was responsible for that. I thought that was somehow going to be taken. The gremlins behind the scenes would do that. Okay. So we certainly didn't get the pediatric vaccines as quickly. And one of the overriding concerns and major objectives of vaccine development is safety first. So that children, being a vulnerable population who are not just little adults, have different kinds of bodies, different physiology, require different doses, maybe dosing intervals that are different. We have to rely on getting data from the adults to be able to establish, okay, are we in the right ballpark with the dose? Are we looking for things we didn't expect that didn't come out of the animal studies or the laboratory studies, so that when we test these in children, we're doing it in the most safe and efficient fashion. And as Dr. Poland said, there are thousands and thousands of adults who are put into these trials, especially for the SARS-CoV-2 vaccine. And in children, we usually don't use as many, with the exception of the rotavirus vaccine trials that he mentioned, because we have got that safety signal from the adults, so we can get them performed, the studies performed in a way that is more efficient when we finally get around to doing it. But there's usually a 1 to 3 year lag behind the adult vaccines, and it's mainly for safety.
And what did we see here with Operation Warp Speed, Dr. Harrison? It seemed like it was a much shorter time frame, maybe 6 months or so before we were starting those pediatric studies?
Well, it is true that if money is not the object, you can get a lot done. So that when you can have 3 shifts running 24/7 all the time, making, refining, producing vaccines, and you pull together all the academic and biopharma resources of a country as big as the United States, you can do these kind of things. It's like getting to the moon. If you put your efforts into it and everything is focused, things can happen in instead of being consecutive, sequential. Most vaccine development is step after step after step, and you make sure the first step works before you do the second step. If you can get the initial information from that first step and start doing things in parallel, then this allows all the same things to happen, but in a shorter time frame. And the same is true with children. And we knew ahead of time from the adult data, and we started recruiting children and their families way before we could start actually giving vaccine in arms or legs, in the case of some young children. And so that process was, it was very exciting to be part of it, and it was very time consuming. There were a lot of people who got less sleep than usual, but the attention to detail, as Dr. Poland said, was very, very strict. And even though it was done in a more rapid time frame as far as the calendar, the amount of hours and effort that went into it probably weren't much different than if it had been spread out and done sequentially over 2 or 3 years.
It's a great point. I mean, that allowed us to really speed up the process by running these in parallel. So before we break down the research, let's define some language you're going to hear us use. We've talked about mRNA, Pfizer, BioNTech, Moderna, are all mRNA vaccines. Dr. Poland, how do mRNA vaccines create an immune response? Do they contain the live virus?
Yeah, it's a question I get a lot. They do not contain any live virus. If we could just use as an example, let's say that SARS-CoV-2 was this pen, which doesn't show up very well, but you can imagine the clicker on the pen. Let's call that the spike or S protein. So we're not giving the whole pen. We're giving the genetic blueprint for the clicker, the spike protein. And that then is able to be taken up by cells in the cytoplasm, not the nucleus of the cell. So it never enters that portion of the cell that contains our own genetic material. And that blueprint is decoded to make spike protein, which is then displayed on the surface of our cells. And our bodies, our immune systems, recognize that S protein as something foreign, and they initiate an immune response. So particular types of immune cells are activated, antibody is produced, long-term memory cells are produced. There's something called an innate immune response that happens within hours. So a broad set of immune responses occur in response to that S protein. So it's a brilliant way of displaying a pathogen-derived antigen. That's a mouthful, but basically that piece of the virus that if we make antibody against it, will neutralize the virus if we were to come in contact with the actual live virus.
Well, great point. And I think we're still learning about that immune response, Dr. Poland, and learning that, those protective antibodies may fade, but then we have these immune cells that now have been programmed and can provide us even longer protection.
Very much so. In fact, what the data would show in adults is that the immune protection against death or very severe disease in somebody with an otherwise normal immune system is very long lasting. How long lasting, we don't yet know. But the protection against lesser gradients of disease, such as infection or mild disease or even asymptomatic infection, that can fade or wane relatively quickly, as it does, just as if you were infected with the virus. So whether we're talking about illness-induced immunity or vaccine-induced immunity, those both wane with time. And that is somewhat of an enigma, a puzzle as to why that occurs with all classes of coronaviruses. Let me just make the point. Dr. Harrison sees it probably more often than I do, but there are things called seasonal coronaviruses. We know about 4 of them, and they are in large part responsible for the colds that we have every winter. Children get infected with them kind of rapid fire. Adults less often, but still do. But the point is we still get infected with them, even though we've been infected with them in prior years. For some reason that we don't entirely understand, the whole class of different coronaviruses does not seem to lead to long-term sterilizing immunity.
And that's a great point. I bring that up to my patients sometimes. The common cold, we don't have a vaccine for it. It drives us crazy every year. And so we're starting to learn lessons from COVID-19, I think, in that there are some things that we are going to have to live with potentially because we only have that short-term immune response that you're talking about. Dr. Harrison, let's switch over. We've talked about mRNA vaccines. Let's talk about now viral vector vaccines. Johnson & Johnson is an example of that. AstraZeneca vaccine is an example of that. What does that mean?
Well, virus vectors are another technology that's been around for a while. Virus vectors have been studied starting in the 1970s. And to understand what viral vectors are, we have to talk a little bit about what viruses themselves are. And there's, in a simplistic approach or thinking process, there are two main parts to viruses. There is the outer shell, you can think of it kind of like a microscopic ping pong ball, that is protective of an internal machinery. And the outer shell has sticky parts on it that allow it to stick to the human cell. Inside is the machinery that allows the virus to make thousands of copies of itself once that machinery enters into a human cell. So a viral vector is, in the case of the SARS-CoV-2 vaccines, a non-SARS-CoV-2, a non-COVID virus, that the shell's been left alone, but the internal machinery has been modified so that the virus can't make thousands of copies of itself, but there are instructions inserted into it that allow that virus to carry those instructions and put them into the human cell. Those instructions cause the human cell to make thousands of copies of the spike protein that Dr. Poland was talking about. So instead of it being an mRNA signal or message, it is a message carried inside this virus, this crippled virus, that allows the human body to make thousands of copies of that spike protein that then are expressed on the cell the same way, and the immune response can occur in the same way. The viral vector is degraded and broken down and is gone within days of being injected. The spike proteins can last up to a couple of weeks to allow an ongoing immune response that's robust. And these viral vector vaccines have also been very effective. At first, the numbers didn't look really good, but as time has gone on, they've held up very well in the protection against severe disease and death, so that we end up with a vaccine that's another clever way of getting the spike protein into the human body. We make an immune response, but we don't have to suffer the infection to get that immunity.
So I think I get it. So that viral vector, it's kind of like what Dr. Stites would refer to, the Darth Vader. It sends in that ship there and attacks those cells in a very stealth fashion. Well, let's talk about, now we've talked about mRNA vaccines, we talked about those viral vectors. Now let's talk about the studies that these went through in order to get to where we are today. The vaccines were approved for COVID-19, went through some very rigorous trials. We describe these as the gold standard. These are randomized, double-blind, placebo-controlled. Why is this considered the gold standard? I'm going to answer my own question, but I'm going to let others weigh in as well. And the reason is, when I do a study that has a shot, and I give you that shot, you're going to feel that shot for sure, and you're going to think that you got the real thing. In fact, a number of years ago, we did a vaccine trial with the flu vaccine. 30% of them thought they got the vaccine, got side effects, thought they got soreness and so on. And the placebo group, exactly 30% as well. So, when you do these trials, the mind is very powerful. And so we have to do it in what we call the gold standard. Double blind, which means I don't know what you're getting, you don't know what you're getting as a participant in a clinical trial. And this double blind, placebo controlled, that second part, that placebo, is so important. It has to be given the same way as the active treatment, whether it be a COVID vaccine or some other treatment that we're using. You have to have a placebo that looks just like it, feels just like it, goes through the syringe just like the other vaccine, because you don't want the person that's given that shot or the person receiving that shot to understand whether or not they're getting the real thing or not. And certainly they can guess, and sometimes not as successful. What about my other colleagues on the panel? What do you see behind these double-blind placebo-controlled trials?
Yeah, Dr. Castro, you explained that very well and helpfully. Let me just attack the randomized part of it. So what randomized means is that once we establish who and who cannot enter the trial, for example, as Dr. Harrison was mentioning with the adult trials, we would say, well, you have to be age 18 and older. You have to be able to understand the risks and benefits and give us your informed consent. Once we have all those criteria, then what happens is randomly, without regard to any other feature of your being, you are randomly assigned to either get the vaccine or to get the placebo. And as you mentioned, the double-blind part of it, the subject doesn't know what they're getting, and the investigator doesn't know what he or she is giving. So there can be no bias in terms of attributing a side effect, for example, preferentially to one arm of the study or another. It also tends to account and level out attributes that we may not know are important. For example, having a high body weight might decrease your antibody response. Having type 2 diabetes, being older rather than younger, all of those things might affect your immune response. By randomly assigning people to either group, we level out and tend to negate those differences. So that's a key piece of running these trials so that we can interpret them, both in terms of protection and in terms of safety in an unbiased way.
Thank you. I have to harken it back to Kansas City and think about, the Kansas City Chiefs and the Buffalo Bills when they were going into that overtime. They had to flip that coin, and thank goodness we got the football. So, it's that flip of the coin that is that randomization. Dr. Harrison.
Well, I was actually going to use the same one, but my memory is of the Bengals and the Chiefs. But flipping the coin is you don't know how it's going to turn out, and it's totally random. But the other thing I think that I like to talk about is that, in using another sports analogy, both of a fan of the Jayhawks and a fan of the Tar Heels can look at the same play in the NCAA tournament, and that play in the mind of the person who is the Jayhawk fan could say, "Hey, that was a foul." And on the North Carolina side, that wasn't a foul. And that's that built-in bias of looking at the same data but getting a different answer. And if you had a fan that was favored neither one, that's a more objective kind of view without bias. And that's what we need in these studies, not to have that inadvertent bias that flavors our interpretation of what we're seeing or hearing.
Well, that's a great addition, because we all have those inherent biases. So let's talk now on Show Me the Science about trial protocol. This is another word that we tend to use, and it's critical in clinical research. When we do these clinical trials, we use this trial protocol. I tell my research coordinators, it's your Bible. It's what you follow, and you follow it to the letter in that regard. Why and what role does diversity play in these trial protocols? Dr. Poland.
Yeah, this is a very important question. It's one we work very hard on. So, but let me interpret the word diversity broadly. So, for example, body weight, age, race, ethnicity, whether one has a certain medical condition or not. All of those are things that can potentially impact the immune response. So you wouldn't want to begin your initial clinical trials, for example, with people that you would think might not respond well to the vaccine. Let's take somebody heavily immunosuppressed from maybe an organ transplant. That's not the group to test the original testing, to test your vaccine in. It's a group you'll get to once you understand safety, once you understand immune response in people who have otherwise normal underlying immune systems. So those are attempts to understand differential vaccine efficacy by important parameters. And of course, when we say diversity, we're mostly probably all thinking race, and that's a critical one. For example, we know that in our minority communities, there's a much higher incidence of what are called comorbid conditions, that is, underlying medical problems. It might be diabetes, it might be smoking, it could be any of a number of medical problems. All of those potentially impact, that is, decrement or decrease the immune response, making them more susceptible to infection and potentially to complications and more severe disease if they were to get infected with SARS-CoV-2. Indeed, that's part of what we have seen during this pandemic, of a higher death rate in our minority communities than in our majority communities. We haven't entirely teased out all the factors involved in them, but some of them involve the underlying medical condition. So it's really important that we understand that, take that into account. And as we learn about that, let me make a point. It actually harkens back to the question Dr. Harrison answered about, why do we start with adults and then kids later? Well, in this pandemic, for this virus, what we very quickly learned is that the older you were, the more likely you would be hospitalized or die. If you were a member of a minority community, your risk of complications or of severe illness and even death were higher than in the majority community. We learned that if you were obese, if you had underlying heart or lung disease, that you would suffer an increased risk of complications. So those were groups of people that it was really important for us to test and know about vaccine safety and vaccine efficacy. Fortunately, we did not see anywhere near the complication rate or the death rate in children, thank God, that we saw in adults. Now that's not to diminish the horrific consequences of what happened to, and Dr. Harrison probably has better numbers, I think around 1,000 US kids or so died during this pandemic. But we're talking about nearly 1 million adults that have died. So it was really important that be tested in adults because they were the ones that were dying of this disease. They were the ones causing the surge demand on the medical system.
Well, it's a great point, and I just want to be clear. You're talking about deaths related to COVID, not to the vaccine itself.
Right.
And so these are clinical trial protocols that tell us how we run these studies, and during those we capture information about how the vaccine is working and what the safety is in regards to that vaccine. Dr. Harrison, tell us a little bit, how do you apply this in pediatrics? Do you follow those same protocols?
Well, the protocols in adults are important because they set the stage for the pediatric protocols. The pediatric protocols are usually written for most vaccines after the data are already in adults, and those tell us how we need to adapt that original protocol to better fit the smaller, less mature children whose immune systems aren't as well developed, who may have somewhat different side effects, who may need different amounts of vaccine given at different intervals. And children, by the way, almost never get long-term immunity without a series of what we call priming doses. Those, if you think about baby shots, where they get the one at 2, 4, and 6 months of age, and then they get another one sometime in their second year before the immunity is long-lasting. And then again, they get another dose when they go to kindergarten. So in children, we know we're going to take a series of initial vaccines to get the protective immunity up and running, and then we have to periodically boost it. And we found out with SARS-CoV-2 that it's a new enough virus that we're all children in a way, and we're going to do the same kind of process where we get those priming doses, the 1 or 2, and then we're going to need boosters periodically. And that's going to be a way of life because we've not had an experience with this kind of coronavirus in our previous years, and so we're all children as far as SARS-CoV-2 is concerned. Now, back to the real answer here is that usually those protocols are informed by the adult protocols. They're parallel, they're templates that have been modified to adapt to children. We follow children sometimes a little bit longer because we know their immune systems can fade faster, and maybe they'll need a booster sooner than we thought. The side effects that we see in adults, we know to look for them more carefully in children, and we add extra questions or extra ways of monitoring for side effects that we're seeing in adults in a child. So those are things that are similar but different. It's like the children's protocols are cousins of the adult protocols.
Well, great explanation. And so I think we now understand about these clinical trial protocols. They tell us what type of patients we're going to enroll. They tell us how we're going to conduct the study. For pediatrics, we're going to sometimes need to adjust that dose of that medication that's being administered as part of that trial protocol. And it's going to help us capture all the information we need to decide, is this working like it should be, and is it safe? Now, those clinical trial protocols go through a number of phases, and we call these trial phases. The FDA basically has set this in the U.S. as 4 different phases. And these represents a series of steps that we take to evaluate any drug or device, but in particular here, this was pertinent for evaluation of the safety and effectiveness of COVID-19 vaccines. So when we talk about phase 1, typically that's the very first time that we're just starting to get it into individuals. Sometimes these are normal individuals. They don't have any underlying disease, but we just want to limit it to maybe 20 or 30 individuals, and we want to test what that immune response is. We want to test what the readout that would be to determine what dose we're going to use for subsequent trials. Often when we start with a new medication or new vaccine, we don't know the exact dose that we need. We have maybe a pretty good estimate based on preclinical studies or studies we've done in animal models, but we need some information in these early phase 1 studies. Once we get that information, we then move into phase 2 studies. Phase 2 studies typically will be less than 100 individuals. These will then allow us start to first get into those at-risk populations that Dr. Poland was mentioned to. It allows us to start to get into patients that have underlying chronic diseases for us to evaluate how does this function within an individual that has underlying comorbidity, as Dr. Poland mentioned, with those underlying chronic diseases. Then phase 3 is really when we launch into that next much larger phase. Typically, these are 1,000 or greater in terms of number of patients. That phase of the study really is going to give us that information about the diversity. How does this work in patients that are thin versus patients that are overweight? How does this work in our African American populations versus our Caucasian populations? And it is so important in the phase 3 that you have those large numbers, so you have sufficient numbers in each one of those categories in phase 3. Those 3 phases are what are required by the FDA for approval of a drug or a device. There is a 4th phase, which I'll turn it over to Dr. Poland to talk about, because it involves a lot of that post-marketing surveillance that he was talking about.
Yeah, thank you, Dr. Castro. I mean, as you mentioned, phase 1 has, let's say, tens of patients. Phase 2 will have hundreds of patients. Phase 3 will have thousands to tens of thousands of subjects in them. Phase 4 is after the vaccine is actually approved and in use. Now we can collect safety and efficacy data across time and in hundreds of thousands to millions of subjects. So let's say that a side effect occurred 1 in a million. You will not know about it during the phase 1, 2, and 3 trials. You don't find out about something that occurs 1 in a million until you have observed millions of people getting the vaccine, so that you can distinguish between what is coincidental or background rates and what is actually caused by the vaccine. So it's a critical piece of the safety and efficacy puzzle, and you can only do it once you have released it. You might ask why. Well, this might surprise people, but on average, the cost to go from the idea of a new vaccine to actually getting it approved in the US is around about $1 billion. So you could not afford to do a clinical trial that involved millions of people. And that's the reason for the phase 4 trials.
Well, thank you for explaining it. So we've covered a lot of information today. I'm sure there are other terms and questions that you may have, or just general COVID questions. You can send those to us in YouTube, Facebook, the Medical News Network, and now Twitter. The links are on your screen. Let's now go over to Dr. Dana Hawkinson, Medical Director of Infection and Prevention Control, Infectious Disease Specialist in our house now. He joins us.
Yeah, hi. Cases today, 9 active infections, 3 in the ICU, 0 on the ventilator, and 35 in that recovery period as well. So overall, the active cases are going down. We know that we have a mix. Half of them are here actually for COVID-type symptoms, and other patients here have possible alternative diagnosis. Again, certainly different from when we saw our Delta and Omicron surges, when the vast majority of those patients were here because of COVID-19, the disease symptoms, and that reason. So overall, cases are going down around the United States. In our community, we haven't really seen that cases or the hospitalizations, which again is more important. As we talk about vaccines, and this was mentioned earlier, we are talking about really trying to prevent that severe disease, that hospitalization. It's not necessarily about blocking infection, but it is to help prevent hospitalization and that whole spectrum of COVID-19, especially the severe disease that leads you to the ICU or to death.
Thank you, Hawkeye. Those numbers look so much better to me. I mean, I looked at those every day, and I was like, oh, good. We are rounding this curve, and let's see what happens with these upcoming variants. But our hope is that this will maintain in those low numbers. Let's move over to our community questions. Jessica, we've been talking a long time. Do we have any questions from the community?
Yes, we sure do. Tanya wants to know, she has a question for our panel. Why are mRNA vaccines more effective against variants?
Great question. Dr. Poland.
That is a great question. Please let me know the answer. I'm being a bit facetious here, but what is happening in kind of fast speed, and so we're still learning here, is what is called the antigenic distance between the Wuhan strain and now the Omicron and the next strain after that, what's called BA2, is increasing. At some point, those vaccines we originally got may not offer very good immunity against the variants that are developing now or will develop in the future. So the mRNA vaccines have done a yeoman's job because they induce really profound depth of immune response, very high levels of antibody. Remember that when we were doing the clinical trials with the mRNA vaccines, we were hoping that these vaccines might be 50 or 60 or maybe even 70% effective. They were 95% effective in preventing symptomatic infection. We then learned that that will fade with time and be reduced further as each new variant develops. So the big question, in fact, while we are having this conversation, the advisory group for the FDA is meeting right now on discussing the whole area of boosters and whether we might need in the future what would be called a variant-focused booster.
Can I add something to that?
Yes, Dr.
My sense, and this is not proven by any stretch, but it is sort of a way to think about it. As you said, the fat particle that this mRNA is encased in when it's given to us stimulates that innate immune system very well and sets the stage for that very profound protection due to the very high levels of antibody that are produced. And as the variants evolve, they are variants, but they're still pretty closely related.
Right.
So it's like, if you look at it in our neighborhood, the O'Connors, there's a lot of kids in the O'Connor family, and they all kind of look alike, but none of them look exactly the same, but you can tell they're O'Connors. And so when these variants come up, they're still O'Connors, but the antibody doesn't stick to the variant as tightly as it did to the original one. So it takes a lot more antibody to glom on and neutralize that variant virus than it did the original virus that was the target of the original vaccines. So they still work, they're just not as efficient. So the boosters increase that antibody again, so there's so much more antibody, it can still overcome this. I'm a younger son that I look a lot like it, but I'm not exactly the one, but I got enough antibody stuck to me, I still can't get anywhere. So there's that relative efficiency of the antibody in relation to how distant that variant is from the original strain that we saw and targeted.
And, you know, the other thing is we do develop some long-term immunity. It's not perfect, but through that innate and adaptive immune response, we develop resident bone marrow cells that remember that they have seen that spike protein and will stimulate immunity if we're infected with anything that looks like that spike protein in the future. But as I think we're both pointing out, how different will that spike protein look over time and with further mutations?
Jess, great question. Additional questions?
Yeah, we sure did. Thank you, Dr. Harrison, by the way, for explaining things in the way even I can understand. Right? So Mike has a question and says it's really good to see Dr. Poland on the broadcast this morning and wants to know if he would share his experience with tinnitus after vaccination and talk about if there's been any research or study into this unique side effect.
Sure, happy to. Let me start by saying a critical mistake that is often made is to assume that temporality is causality. In other words, because something happened to me at one point and then I had a side effect right after that, that it was caused by that. So with that understanding, after my second dose of an mRNA vaccine, I developed within about 90 minutes tinnitus or ringing in my ears. That was not recognized as a side effect at the time. Subsequently, there have been about 15,000 reports to what's called the VAERS system. And now in Europe, for example, there's a warning attached to the J&J vaccine about the possibility of tinnitus. So you might imagine that with some trepidation, I evaluated the data in regard to whether I should get a third dose, that is the first booster. I decided it would be way too ironic if I died of SARS-CoV-2. So I did get my booster, and it slightly worsened it, made it a little higher pitched and a little more persistent. So there's a lot of work ongoing right now to determine did what happened to me and to a lot of other people, is that coincidental? Is it higher than the background rate? We don't know yet, to be very honest and frank, and that's why we follow the scientific method. But a lot of research now getting stimulated into that possibility of a side effect. There's a second part about talking about side effects that's really critical, and here's where wisdom comes in. A lot of people hear about a side effect and they're afraid. What they don't take into account is that one of two things is going to happen to every one of us. We either get the vaccine and are protected from infection, or we don't get our vaccines and we will get infected with a virus this infectious. There's a third group where you got vaccinated and you got infected in one order or another. And so you look and say, well, for all of the side effects that we think we understand with the vaccine, what's that rate of side effects in people who didn't get the vaccine but got infected? And in every case, when we look at this, the rate of side effects or adverse events in people who are infected is far, far higher than in people who might suffer a side effect from a vaccine. Nonetheless, we try to match the right vaccine for the right person when we are practicing our clinical medicine.
Greg, can you tell us and put it in perspective of what the rate of the tinnitus is in relation to the total number of doses that it took to find those cases?
Yeah, you're talking about a minuscule number. Now, nobody knows exactly, because what's called active surveillance, going out and asking everybody who got the vaccine, did this happen to you? That's not how things are done. Rather, it's passive surveillance. Somebody has to realize, this is odd, this happened to me after getting my vaccine. They report it, and that collection of reports then forms a hypothesis. Maybe this is a side effect. And so they investigate using different safety surveillance systems. So, you're looking, the last study that I saw suggested that you might be looking at something as high as a few dozen per million or millions of vaccinees. Higher than that if you don't get the vaccine and get the disease.
Good points. Hawkeye?
Yeah, I'd like to kind of echo what Dr. Poland was saying, just about the infection and the chance of complications to that versus the vaccination. There was a recent MMWR published by the CDC looking at just that, comparing myocarditis and pericarditis in persons who got vaccination versus persons who got the infection. And the people who got the infection had multiple times more risk of having the heart inflammation than those people that got the vaccine as well. So I think that was a very important point that he made. The infection versus the vaccination, and the vaccination continues to be the safest way to gain that immunity.
The other aspect of that will, I think, prove to be terribly important, and again, we're just learning and getting a handle on this, is the long-term consequences of infection. And those appear to be considerable, with 2 to 3% demonstrated shrinkage in gray matter of the brain, with a significantly increased risk of diabetes, of cardiovascular diseases, as Hawk said. These I think are going to be, the next pandemic, if you will, related to the original pandemic, and that is physical and mental health issues as a result of infection with the virus.
Dr. Castro, we've got a lot of really great questions today, so I'm going to just try to maybe quickly get to maybe 2, 3 if we can. So just Jean wants to know quickly what the doctor's opinions are about the mask mandates dropping in public and certainly on airplanes or in airports. What are their thoughts?
I'll just say one real quickly. So it's too soon. It's just too soon to do that. In fact, if you look at the roadmap to recovery, which I thought was well thought out, the number of deaths that we will probably have to live with due to COVID per day is 165. Well, that is about 7 to 8 times lower than the current number of deaths. It's just that our response to those have been blunted because it has dropped so much from what it is. But if the pandemic were just starting today, and I said to you something like 165 cases of polio every day are going to occur in America, America would be panicked. But when I talk about 165 deaths due to COVID, oh well, that's a lot less than it was.
All right, Lynn wants to know if the doctors can tell us who names the variants. Who's responsible for giving out the names?
I believe it's the World Health Organization in concert with, other experts around the world, or other groups who work on the viruses. But I'll defer to our guests and Dr. Castro if they have any other insight into that. But I thought it was the World Health Organization.
Yeah, you're right, Hawk. And what they decided to do is, rather than use a location which sort of unfairly characterizes a geographic area, they're just linearly going through the Greek alphabet with a couple of exceptions.
Yeah.
And the other thing they do is that they add the letters and numbers to be able to tell the one O'Connor from another.
Janet wants to know if Dr. Poland, she wants to know your thoughts on the effectiveness of Evusheld.
Very effective, but in the face of Omicron BA2, the dose is having to be doubled. So one of the consequences of us pretending the pandemic is over and prematurely dropping masks and the safety precautions that we know to do is that these variants continue to arise. And what we've seen is when they arise, they depress the effectiveness of our vaccines, and they sometimes eliminate, if not depress, the effect of our monoclonal antibodies. Evusheld fits into that category for adults. We're now having to give double the dose that we did previously.
Hawkeye, we share kind of our local experience with that?
Yeah, we were giving it. We do feel, just like Dr. Pullen said, it does protect you very well. It is extremely important for those people that are immunosuppressed. But we've also now had to go back because of the FDA issuing the revision to go back and contact those people who got those early doses so that we can give those higher doses.
Castro, this will be the last question, and it's from Lou. He says, you say fade with time. His question is, how is that determined?
Well, I'll give a quick answer. Is it's determined by when we have them come in and get a blood draw. When we do our clinical trials, we do this over a period of time. In our vaccine trial here in Kansas City, we've been evaluating the effectiveness and safety of the AstraZeneca vaccine. We're still following those individuals 2 years out, so we can study what's happening, what's fading over time in terms of those protective antibodies. Others on the panel?
And frankly, the country of Israel has been a place where a lot of our data comes from. Clinically, we can also follow this. So, for example, the recommendation for a second booster, they have a national electronic medical record system where they can capture hospital admissions, deaths due to COVID, et cetera. And what they can tell us is that after X amount of time, we start to see hospitalizations or deaths go up. We give a booster, we start to see them fall down. So we will actually be able to define the time period that these vaccines provide protection for each category of disease.
And I just want to make a plug for the boosters. I got my second booster 2 days ago. I'm at the age where fading is expected. So for people who are in the age group, go get your second booster. I've got a sore arm, and that's about all I got. So boosters are good.
What are those recommendations on the booster, Hawkeye?
Yeah, so right now, as of last week, the FDA and CDC met. ACIP did not meet about this, but FDA and CDC, they followed with the FDA and said that if you are 50 and above and more than 4 months out from your booster, so if you are not immunocompromised, it would be your third dose, then you can. It is okay to go ahead and get a second booster, so a fourth dose. For those immunocompromised, they want you to get a fifth dose, again, if you're more than 4 months out. Again, there continue to be experts who debate this, but I think that the data, and again, we've mentioned Israel, they have been on the forefront of helping provide some of that data. A recent published article in New England Journal of Medicine yesterday did show that those people, and I think mostly they looked at 60 and above, that got that fourth dose did have a reduction in their risk of severe disease compared to those people that just got their third dose as well. So there is continued debate, but I think clinically, if you are in those higher risk factors, especially with age, and you can reduce your risk of going to the hospital or having severe disease even by 5 to 10 percentage points, I think it's worth it because it's not just about the numbers, it's about the individual patients we see every day. And if we can help reduce your risk, I would say it's a good thing to do to get that booster and help reduce that risk of hospitalization and severe disease.
All right. We're going to move on to wrapping this up. This has just been a great discussion. I'm really looking forward to more of Show Me the Science episodes on open mics. Let's get our final thoughts from our guest, Dr. Pullen.
I think the most important thing, and I hope by listening to something like this, the community, lay people realize, wow, there's a lot to this. There's a lot of nuances here. I'm probably not able to simply scan the newspapers or social media and make an informed decision. I would say partner with your trusted medical center, with your trusted healthcare provider, and really understand. Don't make decisions based on fear. Make decisions based on the data. And I hope we've played a small part in doing just that today.
Thank you, Dr. Pullen. Dr. Harrison.
Well, I'd like to make a plug for people getting their children immunized. I know not all of the ages are eligible at this moment, but the use of the pediatric vaccine has been disappointing. We need to protect our children. As Dr. Pullen said, we don't know what the long-term effects of infection are. The children have not been hospitalized or dying, although there is that multi-inflammatory syndrome, MIS-C, in children that does cause a Kawasaki, a heart inflammation, they get hospitalized, end up in the ICU a lot. But that's a very small number. But the majority of children are doing pretty well when they get infected, but we don't know what's going to happen down the road. And protecting them from infection by getting their immunity from a vaccine that is safe in the ages that have been released, and the details with which the efficacy and safety are being looked at are exemplified by the fact that we had to delay approval of that one age group because the dose that was picked wasn't perfect. But getting that vaccine is going to be the best thing you can do for your long-term health of your children.
Great point, Dr. Harrison. Dr. Hawkeye.
Yeah, I'm just going to thank our guests, always so insightful, and I think really help to educate us and our community and really work towards continuing to give that scientific information so that it can be easily digested and understood so that we can, again, keep our community safe, really try to keep everyone safe. So thank you very much.
And I just want to thank our panelists. This is exactly what we need to get this information out there and show me the science. From experts like Dr. Pullen, Dr. Harrison, and Dr. Hawkeye, really sharing this information with you. And they're the latest source. Dr. Hawkeye is quoting the New England Journal of Medicine from yesterday. So, they keep on they really know this data well in terms of sharing that information. Jess, tell us about our guest tomorrow.
I will. Okay, so I have to wrap up the show, though, having just a little bit of fun with Dr. Harrison and one of our viewers who definitely has been hanging on your every word. You're obviously very brilliant, but she says, totally off topic, but does anyone else think that Dr. Harrison resembles rock star John Bon Jovi? And now I can't unsee it. And this is a compliment. I had lots of posters of John Bon Jovi in my bedroom. He's my favorite rock star growing up. So there you go.
Tell my wife.
Yeah, right? See, very, very cool. So I had to pass that on to you. Okay, so Dr. Castro, thanks so much for being with us today. This was such a great conversation. Tomorrow, lots of good fun because it's opening day at the K and we are going to be live out there at the stadium. We've got a great lineup of guests. Plus, all you need to know about heading out to the ballpark, what's changed and what's the same since the last year during this pandemic. So we're going to also tell you how to enroll in another research study, but this one is centered around concussion.
Subscribe to our morning medical update and open mics with Dr. Stites podcasts. Now, everywhere podcasts are available.